Good laboratory practices (GLP) are a set of guidelines and principles that ensure the consistent and reliable conduct of laboratory experiments and research. GLP is particularly important in scientific and research settings where accuracy, reproducibility, and integrity of data are crucial. These practices help maintain high standards of quality control, minimize errors, and promote safety within the laboratory environment. Here are some key aspects of good laboratory practices. 1. Documentation and record keeping. Accurate and detailed documentation is essential in a laboratory. All experimental procedures, observations, measurements, and results should be properly recorded in a laboratory notebook or electronic database. This documentation allows for reproducibility and traceability of experiments. 2. Standard operating procedures, SOPs. Laboratories should have written SOPs for all experimental protocols, equipment operation, and safety procedures. These SOPs provide step-by-step instructions to ensure consistency and uniformity in experimental procedures, reducing variability and errors. 3. Safety precautions. Laboratories must prioritize safety to protect personnel and the environment. This includes wearing appropriate personal protective equipment, PPE, such as lab coats, gloves, safety goggles, and respiratory protection when necessary. Proper handling, storage, and disposal of hazardous chemicals and biological materials are also crucial. 4. Calibration and maintenance of equipment. Regular calibration and maintenance of laboratory equipment, such as balancers, pipettes, spectrophotometers, and centrifuges, are essential to ensure accurate and reliable results. Equipment should be properly maintained, calibrated, and validated according to established procedures. 5. Quality control and quality assurance. Laboratories should have quality control measures in place to monitor the accuracy and reliability of analytical methods and instrumentation. This may involve running control samples, performing quality checks, and participating in proficiency testing programs. Quality assurance activities, such as internal audits and review processes, can help identify and address any deviations or non-compliance with established procedures. 6. Training and competence. Laboratory personnel should receive adequate training in experimental techniques, safety protocols, and the proper use of equipment. Competence should be regularly assessed and documented to ensure that personnel are qualified to perform specific tasks. 7. Data integrity and management. Data integrity is crucial for maintaining the credibility of scientific research. Laboratories should establish protocols for data management, including data entry, storage, backup, and retrieval. Measures should be in place to prevent data tampering, unauthorized access, and loss of data. 8. Environmental controls. Laboratories often have specific requirements for temperature, humidity, lighting, and cleanliness. These environmental factors should be monitored and controlled to ensure they do not adversely affect experimental outcomes or the stability of reagents and samples. 9. Sample handling and storage. Proper handling, labeling, and storage of samples are critical to maintain their integrity and prevent cross-contamination. Laboratories should have appropriate protocols for sample collection, transportation, processing, and storage, including temperature-controlled storage when necessary. 10. Documentation of deviations and incidents. Any deviations from standard procedures or incidents that occur in the laboratory should be promptly documented, investigated, and appropriately addressed. This includes recording any changes made during an experiment, equipment malfunctions, or safety incidents. By following good laboratory practices, laboratories can enhance the reliability, reproducibility, and overall quality of their research and experimental work,